we started with some evolutionary biology and at the beginning of things. So now we want to end with our last lecture that will allow us to look into the future. We are going to find out what is a virtual brain and how can it be used in medicine. So please, for the last time today, warmly welcome Professor Hiba Sheheitli from the University of Minnesota.
Okay, hello everyone. So thank you first for the organizers for inviting me. It's a pleasure to be here in Warsaw with you. I'm not sure that's the future. So just a disclaimer, nowadays when you say virtual brain, maybe you're thinking AI, ChatGPT, robots running around and taking over the world. And that could be an interpretation of what a virtual brain would do. But in the context that I work on, it's not that. It's a virtual brain that doesn't necessarily think or feel or act, but it's more of a mechanistic, mathematical, in silico object that would allow us to understand how neuronal activity emerges from biophysical substrates. Okay, so I hope as we go along, this will make more sense. So don't take out AI, ChatGPT, all these things out of your mind. That's not what the goal of these models is, at least. They could overlap with that kind of work, but that's not it. And I'm currently in Minnesota with neuromodulation in the neurology and interventional psychiatry lab, working on Parkinson's disease and treatment-resistant depression. But this work is actually from my previous group in Marseille at Aix-Marseille University in the Institute de Neuroscience Seminar, who were members of the Human Brain Project over the past 10 years. So before I start, big credits to Victor Girsa and his team, which really are the owners of this idea of the virtual brain, developed over a decade. And I joined them. So epilepsy work was very much mature, and my contribution was kind of to carry that over to Parkinson's disease, neuromodulation, dopamine. So just give credit where it's due. So the outline will be, I'm going to, as I said, use the case study of epilepsy to introduce what the virtual brain is, the neuroinformatic platforms, the assumptions and that go into it, and the tools that it uses. Talk about the case, kind of the first success case in its use in a clinical study for epilepsy surgery. And then I will talk a little bit about the ideas that are required to kind of extend it to modeling the Parkinsonian virtual brain. So what I have in mind when I put together this talk is not to kind of convey a research result or a conclusion or a finding, but more of an introduction to this framework of mathematical modeling to people who are not familiar with it from diverse backgrounds. So it's more about what is the virtual brain, what goes in it, what kind of questions it can help answer. So feel free to interrupt and ask questions at any point all throughout. So I'm saying a mathematical model of the brain, and this is a cartoon from the book by Izikevich, which is a beautiful book on modeling in neuroscience. He says, well, I developed the most realistic neural network, and this guy looks and says, I don't see any neurons here, it's just equations. So we're trying, when you want to present a mathematical model of a living thing, right, there's kind of two approaches. One is that I want to try to capture everything there is, and I want to make it as accurate as possible, and and then you go to supercomputers in order to compute and everything. And the other is, well, even if I do that, it's still a model. And the saying is, amongst mathematical modelers is that all models are wrong, but some are useful. And so no matter how many equations you write, you're still leaving out something. So if that's the case, then the smart question would be, how much can I leave out and gain simplicity while still preserving some core features that are relevant and informative for the phenomenon at question. Okay. So in the case for the brain, your choice of the model is really a matter of scale, because you can think of the brain as many regions made up of many neurons, but really, as John would say, it's a chemical stew, right? So have a chemical stew, it's continuous in space and time, and its activity is highly dimensional and complex, but you can make choices. You can choose to account for all of that, or you can say, what is it in there that's really going to be informative about what I'm trying to model? Okay. So in this kind of like mechanistic biophysical modeling framework, we start at the level of the neuron. That's kind of the, you could go into molecules and synapses, but kind of not into the chemistry of it, more on the electrophysiology, effect of these things on electrophysiology. So you have billions and billions of neurons. You could either try to put that in your model, meaning I have billions of variables, each representing a neuron, or you can say, I'm going to do some coarse graining. I'm going to say that instead of following all the neurons, I'm going to say each bunch of neurons will have a democratic vote, and they're going to be represented by their kind of mean population. That's what you would, so the first level of the neuron is what you would observe if you're really measuring spikes with electrodes that give you the resolution to measure spikes. The mean field level is what you would get when you're measuring what's called local field potentials, right? You put an electrode and you're getting a signal. It's not single spikes, it's kind of the aggregate activity of the population around it and what's entering that region, okay? So you can, at that level, you could think of saying, well, I'm going to have neurons that are either inhibitory or excitatory. So you put some knowledge into the type of neurons, whether when they receive input, they fire or they're inhibited. And then you have a choice of how big do you want your region to be, right? Because the bigger it is, the less complexity to have. And let's say the classical case is that we take you're thinking, I'm going to break up the brain into maybe 60, 70 regions, okay? And each of these regions will be represented by one variable, okay, which is kind of the aggregate activity of the brain. So, and then this is what the general framework of building a virtual brain, according to that approach, would look like. So once you decide to represent your brain as a set of discrete active variables, each representing the activity of a region, okay, first of all, you need to know, so the approach is as such. We build a skeleton, which represents the structure of the brain, the anatomy, and then on the skeleton, this is the skeleton. Each of these red dots is one region, and then how do I place them in space, right? And how do I put the features, the activity of each? I rely on structural data, anatomical data, and then there's standard anatomical parcellations that say, well, this is the boundary between this region and that region, and this comes from histology, from the type of cells you have, the type of connectivity they have, the type of receptors they exhibit, and so on. So the biology tells us how it's reasonable to divide up the brain, right? We take that, and then we have to make a choice about, because we're interested in dynamics, right? The type of question that this approach is most geared towards is that, okay, we said it's not AI, we're not trying to create thinking machines, then what are we trying to do? What we're trying to do is if I have a medical disorder, what's the clinical approach? The clinical approach is that I do some lab tests, right? In that case, I have some functional data, right? I can measure functional MRI, bold activation, or I can measure electrophysiology, EEG, or MEG, or I can have some electrodes and I have local field potentials, right? So I have some signals in the clinic. Now, there's a huge task there to say, for example, what is epilepsy? How do you see it in the signals? What is it in the signals that makes a brain unhealthy versus healthy, right? So this is like a whole field in itself, and that's what mostly medical doctors do, right? They study, and then now they're being helped by data scientists and all these things. But once you have that, once you figure out that I have a certain condition and I have a feature in the data that allows me to characterize this condition versus healthy, then the question is, how can I relate that feature to what I know about how the brain works? Okay, so that's where the modeling work starts. But there's a big task before that, is to know what is it that you want to model, because we're not modeling the brain, right? Otherwise, the equations would just have to be too complex and under constraint, okay? So we need to know what is it that we want this model to exhibit, okay? This is kind of the approach. Once you have that, that's going to guide you in saying what type of activity in time, what type of neural activity, or what features am I interested in on the level of the individual brain regions. So there's a choice in what is it from what you measure actually you want your model to be able to exhibit, and then what's the smartest way to pick my equations in a way that these brain regions can actually exhibit that brain level activity. And once we have that, once we build this kind of avatar, what we end up with is a network of nodes. So the connectivity also comes from the anatomy. There's what's called diffusion tensor imaging, which is just like MRI. It kind of tells us the highways in the brain. It detects myelination. It tells us this region is talking to this region and so on. So that information complements the actual anatomical parcellation, and now you have a network, right? Think of it like a city. You have buildings connected by highways, and the dynamics part, the differential equations, tell me the activity at each of these buildings, and then you make some assumption about how these signals are being transmitted across the brain. So this is the whole connectome-based whole brain model. Connectome is the highways, which comes from DTI, and whole brain because we're breaking up the brain into discrete numbers, and it's dynamics. So I want to be able to simulate. This whole thing will be equations which I can crunch on the computer, and for a given starting point, I can generate synthetic time series which are supposed to be similar to the real time series of neurological measurements. And I'm going to use the case of epilepsy to kind of show you one way of choosing that activity dynamics. What is it that you want to put in your model, and what is the feature that you want your whole brain model to capture? In drug-resistant focal epilepsies, so I think most of you are aware what epilepsy is. Epilepsy is the disease when you have spontaneous seizures in the brain. And the seizures, they manifest as uncontrolled movement of the body, loss of consciousness. They can take various forms, but the neurological mechanism behind it is that suddenly neurons go on overdrive, and the neurons which are supposed to be doing something and communicating with each other in a coordinated way in some region in the brain, suddenly the activity becomes very fast and hyperactive, and then there's a lot of synchrony between the neurons, which doesn't allow them to communicate well. And this heightened synchrony and overactivity manifests in these clinical manifestations. Now, a lot of types of epilepsy can be cured with medication, but some of them, the medication doesn't work. And so as a last resort, the doctors do is they have to go in there and cut out that kind of bad region where the seizure is being, these neurons that are instigating, that are starting the seizure. And the trick is, okay, where is it starting? Okay, which region should we go in and take out? Because it's a big deal to take out brain tissue. So you want to make sure you're only taking out what's really causing the seizure and not anything more. And you want to make sure that you're taking out all the parts that are causing the seizure. So it has to be enough, but not too much. And this has been done for over 50 years. And since then, the technology has evolved a lot in terms of the surgical technology, what we know about the brain and so on. But the success rates of these surgeries have not increased at the same amount. So there's something missing. And that's what the role of the modeling is. But first, this is what the seizure, so this is what the clinician would rely on to actually figure out where the seizure happens. So it's a patient and then now the seizure has started. And over here you can see the neuronal recordings that are being recorded from the brain. And each of these kind of behavioral manifestations is a result of a hyperactivity in a certain region in the brain. So the seizure starts somewhere, and then it spreads to other regions on this network. And usually what the clinicians rely on is this. You can see that this is normal activity and this is epileptic activity. So you have larger amplitude, but also faster oscillations, okay? So for them to make the decision, they need to take into account the manifestation, the clinical manifestation of the seizure, because certain behaviors refer to activation in certain regions. This we know from anatomy and physiology. But there's also some quantitative ways, some statistics on the frequency and amplitude of the EEG. So, and neurosurgeons are special breed. They're really smart people. They have their neural networks that have been trained over decades, and they know how to do this. It's not like they don't have any clue, but it's more of an empirical kind of approach that they learn in their profession. There's no theory behind it. There's no kind of mechanistic explanation for the metrics that they use in the clinic. And so the idea is that can we build a virtual brain which can exhibit normal activity that looks like normal EEG, and then also given certain changes in parameter can exhibit a seizure, which would be a heightened activity in what region that actually gets transmitted throughout the network. Because if we have that, then we know which region, right? If we use the specific patient anatomical data, the MRI and the connectivity, the DTI, and we use the actual, so here there's electrodes implanted in the brains of these patients to record from the suspected regions. So if we constrain our model with that, then the model will tell us which of these virtual nodes, right, was the bad one. And this is kind of what the pipeline would look like for creating a personalized virtual epileptic patient, right? We start with the MRI for the patient that gives us the actual anatomy of the regions. We take the DTI, which gives us the highways. How are they connected, talking to each other? We put in the dynamics on each node, and then we crunch it. We can simulate it. We know, given these parameters, this is what the behavior is, right? And we also use, the idea is that we want to personalize it for the specific patient. So we use the actual data from the implanted electrodes to infer what the parameters of the model would be for every specific patient. And then we present the clinicians, the committee which is planning the surgery, with an estimation. According to the modeling, we suspect that this region, this region, this region have higher propensity for epileptogenicity. Okay. So in terms of the actual math that goes in there, what the virtual brain is doing is basically solving, when we run the software, it's solving a set of differential equations. Okay, so for, I don't know if the audience has seen what differential equations are, but basically we have certain variables which represent the activity of each region, and then we have the rate of change. So we have equations that tell me at every point in time, if the activity in these end regions is this much, then the change will be this much. So you kind of march forward in time by solving these equations. And the equations account for local dynamics, which is, if I take one region alone, what does it do? Does it stay silent? Does it spontaneously oscillate? Does it go to infinity? So this is something we put in the model, what each intrinsic local population does. And then we put a form of coupling, right? If a region receives the signal of another region, how will that affect the local activity of the region? And you crunch these equations. And the virtual brain platform allows you to actually build these equations and crunch them, even if you're not someone who's taken a course in programming, let's say, or numerical integration, or you're not familiar with how to solve differential equations. So the idea is to make it accessible to people who are interested in exploring this tool, but maybe don't have the time or will to invest in learning, numerical methods and programming and so on. It's an open source tool on the website, and it has some already, if you want to explore this, it already has some built-in data, anatomical data, connectivity, and generic models where you can pick the parcellation, how big you want it to be, you take a sample connectivity, and then you could choose some parameters, and you can simulate forward in time and see the activity in the regions. You could also, because the important thing is it's not just what each region is doing. That's not what I'm seeing in the clinic. What I'm seeing is my recordings from EEG or SEEG or fMRI. So I need some kind of forward mapping between the activity of this local population and what would be the resulting EEG or fMRI or SEEG or so on. So this kind of sensor activity, you also have some mathematical models for that, just from physics. And so these forward models are built into the tool so that you can actually visualize or generate synthetic EEG data or synthetic SEEG data, given a certain personalized model. And this is, for example, what it would look like. And because you've used the anatomical atlas, you can actually position the sensors as they were positioned for a specific patient. And then you integrate forward in time, and you can generate synthetic data of what you're seeing in the clinic would be like for this specific model and this specific parameter values. Okay. And the million-dollar question is what kind of local dynamics do you use? Because that's really going to affect your ability to capture the features and also the computational efficiency of the model. And over there, it's really a whole field. It's a zoo of mathematical equations that go across scales and types. You could either start with kind of the most related to biophysics. You start from what you know about a neuron, a Hodgkin-Huxley equation, for example, which has ionic currents and voltages. And you do some mean field, apply some math theory to derive what would be the population activity, right? That's on one end. On the other end of the spectrum is you use phenomenological models. What does that mean? It means I would create an equation with variables that I can't directly relate to the voltage and the current of neuron, but this variable, because of its dynamics, would act the way a neuron would act, right? So it could be silent, it could fire, and so on. And the mathematical theory of dynamical systems gives us these models. The theory allows us to create simpler equations that are phenomenological, but they actually do the job, right? They act, in essence, of course, in a simpler way to the biophysical equations of neurons. And so here it depends on I hope by the end of the talk you'll see, well, how do I pick my model? Which should I use? For the case of epilepsy, the model that we use for the virtual epileptic patients is actually on that end, on the phenomenological end. Why? Because the nature of a seizure is really very striking and has a striking resemblance to types of oscillations that we know from dynamical systems theory, if anyone has studied that. If you look at the SEEG data, the actual signal, the onset of a seizure has specific signatures, features, the way the amplitude and the frequency evolve when the seizure is happening, and the way it actually ends. If you compute some metrics which have to do with the amplitude evolution and the frequency, the interspike interval, then the relationship here will match features that we know from dynamical systems theory. Now, I don't, I'm not going to go into the details of this. This could, need a whole course to study this, but the idea is that dynamical systems theory tells us that if you see a silent object going into oscillation, and if, for example, the amplitude increases as a square root of r, and the frequency is constant, then when you find the equations for your system, your equations will have this form. For example, it will have an x cubed or an x squared or something. So this kind of black magic is rooted in a theory of dynamical systems. So why is this important? It's because if you see this, you go to a dynamic systems textbook and you say, well, I have these features in how the oscillation is. What's the simplest mathematical equation that will give me that? And you will get what's called the normal form. You can use that. So you don't have to go write down equations for neurons and derive the population model. And actually, Victor Gisa and his group analyzed more than 1,000 seizures, and they found if they look for these features of what's called bifurcations, the normal form, there's actually, not all of them are present. There's some dominant types. There are types of how the oscillation is generated that's more dominant than others. So they took that and devised a kind of complex, but not too complex, given that it does a lot of things. It can go into oscillations and out in the same way that what's seen in the SEG. And this is called the epileptor model. It's a set of equations with variables x, y, z, and x2, xy, which are not directly related to the voltage and current and so on, but which when you simulate them, they actually have the dynamical features of a seizure. Okay? And the thing is that there's one parameter in this equation which determines how prone is the local population to generating an oscillation on its own. So if this parameter is small enough, then the local population will stay, silent. There's no amplified oscillations. But if the parameter is above a certain value, it will spontaneously oscillate. Okay. So this is important because then, when we build the virtual brain, and each node we equip it with this model, and we leave this unknown, then we use inference methods to say, if in the clinic I'm measuring this SEEG, and if the model has this form of equations, what value should each region have for the whole system to generate the synthetic SEEG that look like the clinic? And when you have that value, what you end up with is a kind of a heat map for, well, I fit the model to my to the specific ECG of the patient, and the model is telling you that this region has a parameter which corresponds to kind of being very easily pushed over and oscillating. So these are the bad guys. This is what we suspect is the epileptogenicity. Okay. And this approach is already being tested in the clinic. It was tested on retrospective patients, and it's also being tested in prospective patients. And so far, it shows very good overlap. So the patients which the clinicians decided on which region is epileptogenic and did the surgery. And then you look, well, are they seizure-free or not? In the cases where they are seizure-free, the virtual brain matches the clinician's hypothesis. But in the cases where they're not seizure-free, so the clinicians didn't really catch where the regions, you go and you look at what the virtual brain is telling you. In many cases, you see that it's not matching what the clinicians say. There's often additional regions that were not caught by the clinicians. And what's interesting is that sometimes the regions that end up being designated as epileptogenic are regions where you're not recording from. So this is kind of the power of modeling. When you build a mathematical avatar of the whole brain, you can measure and you're limited in measuring from specific areas. The model will give you information about areas which you did not directly measure if it's actually solid and valid. Okay. So now, in the last 5 minutes that I have, I will try to say what is missing in this picture and what can we have to do different to model the Parkinsonian brain. So what is Parkinson's disease? Unlike epilepsy, epilepsy has a very striking, I mean, it was kind of low-hanging fruit because the seizure is a very clear dynamical object. So mathematicians look at that, oh, I know how to model that, and we know exactly epilepsy, what is the feature of clinical feature of epilepsy is that seizure. In Parkinson's disease, the clinical manifestation is a bit more complex. What happens is that we know in epilepsy, it's not clear. Sometimes the epilepsy is caused by a lesion, sometimes it's genetic, sometimes so the etiology is more, it's not that easily related to the framework of the virtual brain. Well, it could be, but that's a choice. In Parkinson's, what happens, the etiology is that you have a small nuclei, the substantia nigra in the brain, that has dopaminergic neurons that produce dopamine that modulates this other nuclei, this striatum, and all these are called the basal ganglia. These very tiny, small, you're talking centimeters, millimeter nuclei in the depth of the brain that control movement and emotion, and it's kind of like have distributed functions, okay? And these set of cells, they die gradually in Parkinson's disease. So you have deprivation of dopamine innervating this specific very small nuclei, okay? However, the effects, even though the problem is so local, you have deficiency of dopamine in this few centimeters in the brain, it manifests in a large scale and distributed network changes across the brain. The way to treat it, there is some medication that would, or dopamine replacement therapies, but usually they don't work well. Why? Because I have dopamine depletion in a very small nuclei in the brain, but I have dopamine receptors in a wide range of other places in the brain. So when you take a medicine, it's not just going to the place where you need it, it's going everywhere else. And so the side effects could become really intolerable. And usually what the clinicians will have to implant an electrode, what's called deep brain stimulation, in those basal ganglia nuclei and specifically electrically interfere with the pathological activity that's resulting from the dopamine depletion. So it's a very invasive treatment, but it works a lot for many patients, but still it's not without side effects, and still it doesn't always work. So the question is, First of all, why does it work? Can we understand why does it work? Can we make it better? What kind of frequency to use? Where exactly to stimulate? And so that's what virtual brain could help address. And the idea is we'll start with the virtual brain framework as we did with the epileptic patient. We can personalize, build the skeleton, but we also need to include the basal ganglia, which is usually very, very hard to see in diffusion tensor imaging. We don't have a lot of details about connectivity, and usually when people do whole brain modeling studies, they leave out the basal ganglia. The focus is on the cortex. So the idea is we have to put it in there, and we have to find appropriate population model, because we're not going to use the epileptor. We could, but it's not very obvious why that would be the obvious choice. And instead of the seizure, what is it that we want our model to exhibit? We said we need some dynamical features. So in Parkinson's, you have this heightened oscillation in the beta range, which is 8 to 20 hertz. It's amplified in the basal ganglia, but it's also its dynamics, how it synchronizes across the basal ganglia and the cortex, that's also disrupted. So you have a local pathology that's manifesting as perturbation in the oscillations, but also the coordination between the oscillations across the brain. And in order to do that, we say, okay, if I want to model it, I need to have a model with normal dopamine concentration and a model in which dopamine was depleted. That's kind of the most intuitive way. But the question is, what does dopamine do? How do I put it in my equations? And the thing is that dopamine is a classical neurotransmitter. So what it does is that if a cell receives dopamine, it might not be affected at all. It's not going to fire or be silenced. It's not like the actual, like AMPA or GABA, which actually cause the cells to fire. Neuromodulators, what it does is that if a cell receives dopamine, it's going to change how it responds to the input it receives. Okay, that's the idea of a neuromodulator. Yep. And so that was the idea, that we need to put in the basal ganglia structure, which is, if you start studying basal ganglia electrophysiology, you will see that unlike the cortex, where usually, you have diversity and everything, but usually you could maybe model the cortex as being homogeneous in terms of its firing rate and so on. But the basal ganglia, we know that you have these nuclei, STN, GPE, GPI, which have, some of them fire with very high frequencies. They have bursts and pauses. So they exhibit a very rich dynamics in their firings. Okay. So the challenge is, can I find a very simple model that can allow me to capture bursting and oscillation on low and high frequency and so on? And people have tried to model the basal ganglia using single neurons, single neuron models. And you can see that this is what single neurons do, so very qualitatively different type of neurons. But if we want to embed the basal ganglia in a virtual brain, we don't want to be on the single neuron level. So you need to make some assumptions to say, I need to replace this bunch of neurons which acts like this with a simpler equation that I can couple to the whole cortex and the whole brain. And to do that, I need a kind of what's called a mean field model that is versatile enough to exhibit all these properties. And here I'm gonna go fast over the math, so don't worry too much if you don't capture all what the equations are saying. But the idea here is that we didn't go fully phenomenologically, but we started with a very simple model of a neuron, which is based also on bifurcation theory. It's called the Zikiewicz model, or quadratic integrate and fire neuron. So I have two equations for a neuron, the voltage and a slow current. And then I add this firing mechanism that if my voltage passes a peak, I will reset it. So that's how I get away with not having to have more equations. And this model, for different parameter values, can actually exhibit all the firing properties. Okay, so for anyone interested in models of neurons and simple models of neurons, then classical work of Zikiewicz is really a masterpiece and very easily accessible. And then what motivates us more to use this model is that Humphreys in the seminal paper showed that you can fit the response of the striatal neurons in the basal ganglia, can fit the response to dopamine using this simple Izikevich model. So he fit kind of like in vitro data to this model, and he showed you can do that by just a simple scaling. You can say, well, I have this model, and then the dopamine concentration it receives is going to scale how intensely does it respond to its inputs. And if you want to extend that to the mean field level, instead of the epileptor, now I have a mean field model, which through mathematical derivation, you can go from a single neuron to say, if I have a population that's coupled, what would the equations of the mean firing rate, the average firing rate of the neurons be like? And this is what the mean field model would give you. And it's tracking the mean firing rate, the mean membrane potential of the population, the mean slow currents, and here the conductances, how open or closed are the synapses. So again, don't worry too much about the math, but the idea is that you have a model that's simple enough, but not phenomenological. You can still relate it to firing rates and membrane potential. That's good, because if you measure local field potentials, you can relate them to the synaptic currents. And the dopamine effect now enters as a scaling of the coupling between the regions. So remember, we said the coupling comes from the highways, the DTI. So now the neuromodulation enters as a modulation of those streets and highways. You know, some of them are going to become more effective, and some are going to be less effective. And yeah, I'll just show you, you know, just the diversity of what this population model can do. For different parameters, you can have all these different oscillations. And that's important because, as we said, the Parkinsonian pathology is really one core feature is disruption in the beta oscillations in different ranges. So that gives you this kind of versatility that you can tune. And finally, I'll skip over this dopamine. And now, instead of just having one network, you have the regions and just the DTI connections. With this model, you can have your regions and you can specify the type of connections. The synaptic conductance are there. Is it inhibitory? Is it excitatory? You can also specify your dopaminergic connections. So you kind of have embedded connectivities that you can have in there, and then experiment and test what is the effect of disruptions in all of these. And in a simple example, you'd see that here, you can implement that in the virtual brain. Here, we haven't put in the whole cortex yet. Cortex is just being represented as one population, but just to kind of show what the proof of concept for the model, you put the complex connectivity of the basal ganglia, which is inhibitory, which is excitatory. Where is the dopamine happening? Where is the dopamine entering in the picture to modulate the connectivity? And for certain levels of dopamine, you get something like that, which is more like the healthy state. And with dopamine depletion, you can see this kind of exhibiting of pathological oscillations. That's kind of the biomarker of Parkinson's disease, which appears here in the Fourier, in the power spectrum. In the different nuclei, you see the kind of 30 hertz beta oscillation. But again, this just to kind of, the bottom line is this, with a smart choice, by using dynamical systems theory, by using what we know from mean field modeling and the etiology of the disease, we can create an avatar, an in silico in virtual object, in which we can use to input patient-specific data, the anatomy, the connectivity, and also the functional EEG fMRI, and input a hypothesis about the etiology. We can say, well, this connection, the dopaminergic connection, is what I'm interested in inferring or seeing the effect of, and then use it to help predict or understand how to interfere on the brain. And that's it. I will stop here. And if you go to these papers, you'll see the whole team that was behind Virtual Brain, more about the clinical trial that is being done for epilepsy and the model development for the Parkinsonian. So thank you. Sorry I went over time. Okay.
And so, well, thank you so much for this talk. And now we are open to questions. Any questions?
So I want to ask if you are familiar with the Mike Johnson work. I mainly have this one statement that he stated in my mind, that to every conscious experience there is a corresponding mathematical object that is isomorphic to its phenomenology. And basically virtual brain seems to me exactly like that mathematical object that we are trying to model. Would you say it's...
First of all, I'm not familiar with the author, but that sounds beautiful. So thank you for sharing. Second, I'm not a pure mathematician. So isomorphic, are you a mathematician?
Not really.
Okay, so I have a good idea what it might mean. And I mean, I don't know if I would use this formal mathematical language to describe virtual brain, because the brain, one of its, the challenges and one of the key to its powers is degeneracy and redundancy, right? So I don't know if you're saying that there's a one-to-one mapping in a sense,
Maybe
or that
maybe in a perfect word, in a perfect...
Yeah.
I understand that we are doing some calculations and trying to simplify the model to make it easier to compute. And yeah, but basically
it's a kind of abstraction, right? And the idea is that the model is kind of defined, for me, okay, though you can define a model as a set of mathematical equations, right? But also part of the definition is that a model is only really takes its own identity when you say, what is it supposed to do? And for me, it's supposed to help you understand something, and it should give you some informative or predictive power, not necessarily both, right? Different types of model could do both or either. And what I'm saying is that I'm observing something scientifically, I'm making a scientific observation, and I'm trying to abstract into mathematical world. And in that mathematical world, I have, as you said, mathematical objects that I could say they're kind of the shadow, the mathematical shadow of this physical thing that I'm observing. And how do I convince myself that they're a reliable shadow? It's by saying, can I map processes? Can I understand something about processes in the physical world by studying the behavior of that shadow in abstract mathematical space? So that's what these kind of mechanistic models versus, let's say, machine learning models, where it's mathematics, but you're not interested in the abstraction per se. You're interested in the actual outcome of the mathematical process. Whereas here, when you do mechanistic models or dynamical systems, a core part of the work is this abstraction state when you're actually mapping a behavior or a thought or an emotion. But the challenge, you could do that, but the challenge is we don't have good enough knowledge of how these higher level notions map onto neuronal recordings. That's why I stick with medicine, because in medicine, the pathology usually manifests in a clinical biomarker in neurons. When you go to cognition, I mean, it's hard enough to define emotion and feeling in semantic space and then map it to how do you see an emotion in EEG. There's a lot of work, but it's not as, let's say, conclusive as when you have a pathology that takes these notions to extremes.
Thank you for clarifying here.
Bye.
Okay, any more questions?
Okay, hi. Thank you for the talk. And I wanted to ask you, like, in regard of what my colleague here said, and what you said actually, where in machine learning we have no real abstraction, we don't understand what's going on, but we have a beautiful outcome. In this virtual brain, we have abstraction, but we don't really have an outcome yet. Will we ever? Do you think it's possible that the models will be so accurate that we could rely on the outcome of those equations like fully? And if it will happen,
Wait, you're posing 3 questions, and my working memory space is
okay.
saturating. So I'll try to address this, and then if you have more. The first was that machine learning is focused on outcome and not abstraction. Now, that's not purely accurate, because sometimes machine learning is used to abstract. You have what's called autoencoders, for example, right? You could use them to extract, let's say, lower dimensional spaces of your data or so on, but kind of in a different structure without this kind of mechanistic, like you could think there's no mechanistic knowledge coming from the science of something usually, even though there's now also like physics-informed machine learning and so on. So the 2 fields are interacting a lot, and these exist, but in the most first leading order, let's say approximation, you could think of them as different, but they do meet. Okay. The second one is that you have abstraction but no outcome. That's a bit too harsh, because there are some outcomes and they're showing utility, right? They are used, even if you're not using them to make medical decisions per se. At this point, they are used also to gain understanding and form hypotheses that could guide your experiments and so on. And the third one, will they meet? Was that the third one?
Yes, and if it's gonna happen, is it gonna happen in our lifetimes?
Yes, and actually they are meeting. If you follow the work of Victor Giersa, I left the group now. We're still in touch, but I'm not hands-on with the projects in the. His new big project is called the Virtual Brain Twin. So even taking the personalization to a next level. And over there, it's more ambitious than here. Over here, we said by going from the microscopic to the mesoscopic, which is the region, we're going to use either phenomenological models or kind of like reduced or models from theory. What they're doing there is, in the specific case of schizophrenia, is trying to use machine learning to bridge the scales. So they do very extensive, they go to the chemicals too that John is talking about. They do molecular simulations of the effect of medication on the cell and the molecular level. That's one team that does that. And then they develop neural networks, differential equations that are based on neural networks to model that input-output of that detailed by microscopic. And then that machine learning product, they take and they use as one node in the mechanistic virtual brain. So that's kind of the next 5 years or maybe more projects called the Virtual Brain Twin, which you could learn more about if you go to just Google Virtual Brain Twin. You'll see that. So that's where kind of like, there's different approaches. Yeah, if you want to be very empirical, then you have to use all the tools you have, and that's what's happening.
I see. Thank you.
Okay. Do we have any more questions?
Thank you for the talk. Because the math mostly went over my head completely.
That's okay.
Understandable. All respect due, right? But I wanted to say that I think I don't clearly understand how would this approach be used in diagnosis or actually treatment in Parkinson disorder. as it was in epilepsy? Because as you said, in epilepsy is straightforward. You get a localized matrix that you try to surgically remove, but here you know already which structure is responsible for the major disruption in the network. But then what would give us in diagnostic or treatment to have a model that tells us about activities in those different mesoscale region activity, which is also inferred and looked at through some neuroimaging.
So I didn't answer. It's okay that you didn't understand because I didn't address it fully. So it's
okay.
For the sake of time. So I only said first the motivation, then okay, DBS works but not so well, so let's try to model to make it better, right? And that's not very self-explanatory. So there's two ways you could use. If you have, the challenge is, can I build a Parkinsonian virtual brain? In epilepsy, as I said, there's the big major task, which is find your feature in the data and then find the model that allows you to interrogate that. In Parkinson's disease, I'm actually working on both in parallel because the feature itself is still, there's no strong consensus about it. As I said, the beta is like the most obvious, but it's not conclusive. Sometimes you could get relief of symptoms without the beta going away, or you can get bad state without the beta. So it's not like, bulletproof as a biomarker. Second of all, we're only starting to understand the disruption on a whole brain level. Okay, and there's, in my lab in Minnesota, we do non-human primate models, Parkinsonian models, to actually figure out if really, is it just the whole story? It's that this small region that's disrupted? And the answer is right now, probably not. Because so in the monkeys, in the humans, you only have recordings from the DBS, which is just the region inside. But the purpose of the monkey model is that let's try to measure everywhere we can. So we're measuring with electrodes in the cortex and several places in the basal ganglia at once, and trying to first characterize the dynamical feature that makes a brain Parkinsonian. And having a model that kind of already matches what you already know, which is incomplete, would most likely allow you to explore what is it that I'm missing, that I'm still not measuring or still not thinking of. So it kind of guides you to know what to go and look for. Just like in epilepsy, you're fitting the model to data from certain region, but the model would tell you that there's this other region here which you're not observing that might be, because you're trying to capture the system dependencies using the model. So one, it could give you explanatory power to guide your clinical work. What is it that I should look on? The second, the more kind of applied thing is that to guide DBS, should we stimulate with a high, right now you stimulate with a high frequency continuously on. And this has a battery, the DBS device, and it's a challenge to replace the battery and so on. So if you don't need to be on all the time, there's interest not to be on. So now there's new stimulation paradigms based on kind of modeling and experiments in the monkey, which tells you, well, it's enough to stimulate, let's say, for a few seconds and then off, a few seconds and off. Okay, what pattern is optimal? There's also burst stimulation. There's coordinated reset stimulation. So there's a lot of stimulation paradigms that are being explored, and they're being explored mostly empirically by trying them and testing them. And if you had a virtual avatar of a Parkinsonian brain, then you would be able to explore a lot the effects of stimulation and different paradigms of stimulation in a much faster or more comprehensive way that you can do in the clinic or the monkey model.
Thank you.
So unfortunately, we do not have more time for any more questions. And this concludes the last Q&A session of today. As we draw this seminar to a close, we would like to once again thank our esteemed guests for the phenomenal lectures, and to all members of the audience who have joined us today and actively participated in the Q&A sessions. If you'd like to revisit the lectures from today, they will be uploaded online and linked on our Facebook event page. At the same time, we'd like to follow the future events organized by Nencki Open Lab initiative, as well as the Neuroinformatics and Neurobiology Student Scientific Organization. We look forward to seeing you again. Thank you.